Welcome, integrated STEM students. Today we'll be looking at one of the STEM areas, right? It was science before, now it's technology, so we're getting into technology. And the subject area that I wanted to focus on, kind of to lead up to the integrated STEM project for our purposes, is microscopy. So that'll build off of a little bit of the fluorescence materials that we went through last week. So microscopy is perhaps where I'm most fond of working in the biological world is using different types of microscopy. So for a long time, I've been really interested in the subject area. Um, so I'll give a little bit of background of microscopy and some of the uses and some of the variations. So microscopy is a technique that allows us to visualize matter on scales that are not visible with the unaided eye. So two of the most common forms of microscopy that are used are light microscopy or optical microscopy and electron microscopy. So it's kind of using electromagnetic waves or properties within the visual light spectrum, but then also those exhibited by electrons depending on the type of microscopy being employed. So light microscopy uses visible light photons, right? That's between 400 and 700 nanometers on that Roy G. Biv spectrum, whereas electron microscopy behaves in a lot of interesting and similar ways. It's just instead of using visible light and photons, we're using electrons. And the key here is that electrons have a much smaller wavelength. So some examples of these two types of microscopy at play could be using fruit flies, right? So I've actually had terrible fruit fly infestation in my own kitchen this year, constantly battling these little beasts. I'm trying to keep them at bay. So they're common in our households, in my household in particular, but also a really useful research tool. They're useful for studying development, to looking at the way that organ systems and their organs and their architecture are laid in terms of their framework and foundations over the course of development. But then also, if you're asking questions about disease, really basic questions, for instance, these can be a really useful organism because they're so manipulatable at the genetic level and easy to kind of track in terms of genetic lineages and knocking out gene functions and that type of thing. So they're really useful for biology in general. And are frequently the topic of studies, right? So we've got to understand how these small animals and their even smaller organs and their even smaller cells and proteins, how they're behaving in the paradigm of some sort of a study, what kind of changes are being impacted by variables and that type of thing. So we need help to see these little beasts and their organs and cells, right? So this is where we can use microscopy. So light microscopy, I've carefully picked two different types of each here. So in the upper left, you see a fruit fly. So this is clearly using an aid to be able to visualize them with greater resolution than we would typically with our eye, right? And there's a scale bar there. All of them have a scale bar, so you can compare if you'd like. But this type of microscopy would be like a stereo microscope or a dissecting microscope are the common types of terms for the type of instrument used for this magnification and visualization. And you'll note that the light doesn't need to pass through the specimen in order to use this technique, right? Light here is being cast down from on top of the fly, and the light is striking the fly, and the light is striking the leaf, right? And then we're visualizing that through a lens. On the lower left, this is also light microscopy, and this is part of the eye of the fruit fly, So they have compound eyes, ommatidia. So their visual systems bear some similarities to ours in terms of visual pigments or proteins that are specific to certain wavelengths and so forth. But the structure of their eye is really different. But on the lower left, this material was stained to give rise to some variation in pigments to visual eyes. The magnification is much higher, right? So the scale bar in the upper left is a millimeter, whereas a scale bar in the lower left is 1/50th of that, right? 20 micrometers. So just very crude ballparking here. It's about 50 times as powerful as the whole fruit fly, looking at just these kind of structures within the eye. But importantly, that image on the lower left, light passes through this specimen, right? So this had to be a fruit fly that was collected, then preserved, then sectioned, almost like a deli slicer, except on a much, much finer scale, to be able to get really thin sections that light could pass through. And with that careful preparation, right, that allows us to use not a dissecting or stereo microscope in the upper left image, but instead a compound microscope, which is the one that you would usually find in a teaching lab where it has a stage that can handle some sort of a slide, right? And the slide usually has some sort of transparent specimen or tissue on it. So the left, upper left and lower left, those are light microscopy. Now on the right, we have electron microscopy. So I've carefully kind of kept some consistency in the way the panels are arranged because there's similarities in the strategies, kind of the technical strategies. Because on the upper right, this is electron microscopy that specifically it's called scanning electron microscopy. And similar to the light microscopy just left of it of the whole fruit fly, it actually behaves in a really similar way. Except instead of a lamp throwing photons down on the fruit fly and the leaf, right on the upper right, we have a filament or a source that's casting electrons down. And then you have a detector that's collecting the electrons as they bounce off of the specimen. And then the lower right is what we call transmission electron microscopy. And just like to the lower left, light is going through that ommatidia specimen. To the lower right, electrons are going through that ommatidia specimen. So both of them require a very thin section, not always, but oftentimes preserved. And then just on the lower left, where there was kind of histological stains to bring out the blues and the tans, electrons would generally just pass right through a thin specimen. So normally you have to apply like heavy metals and different types of stains that'll make the regions variable in how electron dense they are. So maybe you'll use a stain that'll stick to lipids, and that stain is very electron dense, some sort of a metal that's sticking to those regions. That'll make it harder for electrons to get through with the technique and make those areas look darker as less electrons pass through. We'll talk a little more about that. Okay, so light microscopy compared to electron microscopy. Light microscopy, upper left, is a stereo microscope with photons bouncing off of it. Lower left, photons passing through it. Upper right, electrons bouncing off of a specimen. Lower right, electrons passing through it. One kind of thing to make note is with electron microscopy, where you see these two types of images that are the same image. Yeah, they're the same image. One of them has been pseudo-colored, and that's not that uncommon, where you see these scanning electron microscope images, which are always very visually appearing, you know, the bizarreness of insects shown and so forth. But they're naturally only variations in signal, so they're naturally only grayscale, white to black. If you ever see any colors, which sometimes people take the artistic license or liberties to do, like on magazine covers and things, Those have been pseudo-colored. So they've just come in and artifactually applied colors arbitrarily because there's no color at that scale of the electron wavelength. Right? We're way below the visual spectrum. So any color is just artistic. So where does light microscopy come from? So we can go back and look at a little bit of historical context. So Robert Hooke was a English polymath who had created some of the first examples or use of lenses to magnify the biological world. Right? One of the things that he observed first were cork cells. Right? So these were sample preps where the cell walls had the cellulose and the integrity, where they outlasted all the other cellular material. But it occurred to him that they looked a bit like a cell in a monastery where monks go for some solitary kind of meditation. So he thought they looked like these solitary kind of confined rooms. So that's where they got the word cell, because it actually appeared like those types of rooms. And then this led shortly thereafter to Anton van Leeuwenhoek, who also used a very early crude type of microscope. And you can see on the left, there's a knob, the upper knob with kind of a little like nail coming out of the top. That's where you would put your specimen, because just to the left of that, you can see this little lens, circular lens, almost like an eye, right, embedded in that brass plate. So that would allow for the magnification. But he was the first one to look at single-celled organisms. And in this particular paper that he published from 1683, this is a description. It's a great paper. I've read it. It's pretty short. There's a link if you want to read it. Looks at animalcules, or these little animals that he could only see with the aid of a microscope. And he collected them from folks about on the street by actually scraping kind of that white substance that you can scrape off your teeth, calling it the scurf of the teeth. And of course, our oral cavity is just churning with bacteria. But nobody had ever seen them before. Nobody had seen kind of pulling back the veil on this microscopic world. And this was the first instance where these little live animals or structured could be seen in the scurf of the teeth. And his crude kind of scientific approach was to look at different people in different strata of the social hierarchy and look to see if there was variation in the animals on the scurf of the teeth. So this is the actual paper here. So though my teeth are kept usually very clean, nevertheless, when I view them in a magnifying glass, I find growing between them a little white matter as thick as wetted flour. In this substance, though I could not perceive any motion, I judged there might probably be living creatures. And he goes on to look in the scurf and find some of these tiny little beasts that we know of today as bacteria. He also went on to look at and collect other cells like spermatozoa. So he observed and illustrated different types of representations of visual interpretations of sperm. And this, just kind of an interesting aside, led years later to the left here of wondering how these cells, these sperm cells, how they can possibly be involved in the generation of life, right? So it was thought, well, surely there must be a tiny little human being in the head of each sperm. So this was pre-formation, where the thought was that within each sperm cell exists a tiny preformed human, and that's the way sexual reproduction works. Just an interesting aside. Also interesting to think, without the luxury of accurate assumptions or information, and trying to come up with hypotheses of the way things work, it's always kind of interesting to put yourself back in time from their perspectives. So we can look now at some of the different types of light microscopy, right? So light microscopy uses lenses with light passing through the lenses to influence the path of the light. And in that way, give us this idea of magnification, magnifying something that's below what can be perceived with the unaided eye into something that can be perceived. There's lots of different flavors of light microscopy. So bright field is the one that we've all used, right? So that's with a standard compound light microscope. There's ways that we can influence the path of light before or after it's passing through a sample to bring out contrast. And two of those approaches are phase contrast and Nemarsky differential interference contrast, which go a little bit beyond the scope of this presentation. But a lot of biological tissues and cells are transparent, and it's difficult to really see anything, right, that's perfectly transparent. So one strategy is to apply stains, right? And the other one is to try to influence a path of light to bring out contrast where possible. And that's what those different approaches will do, and we'll look at examples. Fluorescence microscopy uses fluorescent probes like we talked about last week. Super resolution microscopy is something I've had the luxury of being able to use. Within the last decade or so, 15 years, it's starting to move more into the world of research where standard labs can have access to it. They're really expensive instruments, but places like UMass Amherst or UMass Worcester, the medical school there, they have super resolution microscopes. And they use a variation on standard microscopy where the samples can actually be manipulated, and then high-power computer can back interpret that manipulation to break the limits of resolution that we haven't been able to break since Robert Hooke, right, for like hundreds of years. Light's been light. But now with computers, we can kind of use some tricks to be able to break the limits of resolution that we couldn't before with light microscopy. So I wanted to show the distinction here because we looked at the fruit fly as the fly, right? But then we also looked at the section of the Yomatydia. So that would be sectioning with a compound microscope, right? Compound light microscope is called that because it has multiple lenses. And then the stereo microscope has this like three-dimensional view. Doesn't need to be transparent. You could throw a sandwich under there, throw whatever you want under the microscope, right? And you can use it to visualize increased magnification. So I want to be careful with throwing around terms like magnification without being thoughtful about it. Because an example I can use is if you're using your phone to take a picture, right, and then you look at that picture on your phone, it looks great. Right? But then if you zoom in, it very quickly becomes grainy, where you can't really interpret anything. You're magnifying it, right? You're making things bigger, but you're not increasing the resolution. Another way to perhaps think about that is if you took a picture and wanted to save some memory, so you saved it at low resolution, right? It might look fine on your phone, but if you were to throw that on a projector and project it up on a wall, right, without more resolution, it would just look grainy and terrible, right? So magnification and resolution are different. In this picture to the left, we're using light microscopy for 1,000-fold magnification, which could be like a standard 100 times objective with the 10 times ocular lens would be a reasonable type of setup for 1,000-fold magnification. Is something that can be done pretty easily with light microscopy. Now, you could blow that up 27 times just digitally. And that's kind of shown by the middle example here of what that might look like, because it would just look, it looks bigger with the region that you're looking at, but without an increase in resolution, it doesn't yield any more information. What you can really do with electron microscopy is you can get much better resolution. So there, in that case, to the right, you have 27,000 times magnification, but still high resolution, so a lot more information. And there you can see those stacked membranes right in the middle kind of jump out at me as clearly Golgi, so the organelles can be visualized. Normally with light microscopy, you're not seeing things like Golgi stuff. You're not seeing mitochondria or the membranes within mitochondria without some additional types of approaches, right? But with electron microscopy, that's totally doable just in terms of magnification. So what is resolution? Let's think about that a little more. So resolution is the idea that you can have 2 points. To the extent that you can differentiate between these 2 points or resolve them, that's resolution. So how close can 2 points be where you can actually see that they are still 2 points, right? If you were to put it another way, if you were to take a marker and on a wall do 2 little dots about 1 millimeter apart away from each other, right? You could do that with a marker right up next to the wall. As you start to step back away, right, what you might initially see as 2 points, right, would start to kind of blur together and become one. So you've lost that resolution. Well, lenses can get in and can resolve specific points in space, and we can calculate that based on the type of objective that we're using. So how do we resolve stuff? If you're in a teaching lab and you have microscopes, right, you'll probably have microscopes, or for sure in teaching labs that you've been in before, you've encountered this idea of microscope objectives on a turret, right? And that's shown on the right, pretty standard. Usually they come with a 4 times objective, the scanning objective, and then a 10 times, and then a 20 and or 40 times objective. Those are pretty standard. So this is the power of magnification of these different objectives. Keep in mind with the illustration in the center, right, when we're talking about compound microscopes, there's also going to be the eyepieces or the ocular lenses. Those are usually 10 times. So it's really, when we're thinking about total magnification, if you're using that 4 times objective, that's fine. But if you're looking through the microscope, there's also the 10 times ocular, which doesn't change no matter what objective you're using, right? So the real kind of realized magnification there is 40 times. I have these lower objectives, 63 and 100, because sometimes they're on standard teaching microscopes, for sure in research grade. But as the objectives get higher magnification, they tend to switch to oil immersion, where you use oil on your slide, on your sample, that creates the interface between your sample and the lens. And we'll look at why that is. So to get more to the heart of resolution, keep in mind that objectives have different magnification strengths. So what's shown in the picture here are 3 different objectives. You may have experienced the issue that you have with different objectives, that the more powerful you get, the closer the objective comes to your sample. So I'm going to back up a little bit here. You'll note that on the panel, the objectives have different lengths. And the red one kind of to the back, with the red ring on it, that objective is kind of short and stubby. That is the scanning objective, the 4 times objective. And the working distance tends to be pretty big. and would be roughly comparable like to the objective to the left, right? So if your sample is the blue horizontal line, there's a lot of space and breathing room between the objective lens and your sample. So that might be representative of like a 4x scanning objective or one of the lower power objectives. As you increase objective strength, your objective tends to have a smaller working distance and get closer to the sample. And this is one reason why, as a teacher, we always tell our students to start with the scanning objective, the 4x, the weak one. Because if you start with a higher one, your objective and sample might crash into each other as you're trying to switch slides. So give yourself some space to work. So start with that lower power objective. Now, as you increase the objective strengths, you also increase the angle from your sample to the lens. And I think about this almost like a book opening up, right? Where if you look at A to the left, your book is almost closed and the angle is relatively small. But as you decrease that distance to your sample, the book kind of opens up and that angle becomes higher. That will be related to what we really care about, and that's the numerical aperture, the NA. So objectives are going to have an NA, a numerical aperture, okay? And that is, if you look at this angle here, like the book, like the yellow cone, right? This angle here, or the mu, right, is 1/2 of the angular aperture, right? And that's also what we use to determine our numerical aperture, shown on the bottom as that equation. So your numerical aperture, your NA, is going to go up as your objective becomes stronger, right? Because if you look at the figures, as you move to the right, your objective becomes stronger. And as you move to the right, that angle becomes larger, right? So as that angle becomes larger, your NA also becomes larger, right? And then the illustrator here who's made this figure has also thrown up there, right, the way that the mu, or that angle, is tied to the NA. So that's pretty useful there. Now there's one other different factor to think about when you're looking at this, and that's that N. And N is the refractive index. And we won't go into too much detail here, but I'll tell you that the refractive index is the way that light behaves when it's moving between two different materials. And you've experienced this if you've had your legs dipped in the pool and they look like an illusion of bending in weird directions. It's because as light moves, right, between materials within the water and crosses over the interface between the water and the air, these have material properties that are different, water and air, and the light's going to behave different as it moves across that interface. And that's a reflection of the differences in or the refractive index moving between those two materials. And it's also why, right, not all objectives just use air. So I'm going to go back, right, because if you look at that equation on the bottom, right, I think it's pretty straightforward that the numerical aperture, or this kind of number that we care about in terms of objectives, it goes up as the angle goes up and as a working distance goes down. That's fine. But the numerical aperture is also a function of N, the refractive index. And as refractive index goes up, right, so does the numerical aperture. And the refractive index of air or water or oil increases, which is why we can get a bigger NA when we use oil at the interface there, and why some objectives you just use oil with, because it's going to have a higher N and to be able to increase that numerical aperture. Right. So here are some standard objectives, right? So we've seen 10 times, 20 times, 40 times, 100 times, right? These are on most microscopes, right? You can also see the numerical aperture goes up, right? And that's because, like we looked at, the working distance goes down and that objective goes down, and the focal point kind of goes down. focal distance. But we've also seen that you can influence the medium that you're using there. So you could use, instead of just air as it exists, right, you could add oil, because oil at the interface would have a higher refractive index and would help to bring up that Na2. Fortunately, all of that is usually written on the objectives. And so this is the same picture and panel from one of the earlier slides. You'll notice that they're different lengths, right? And that's because they're different powers. And you can see the different powers written there. I see 4 for the red, right? 10 for the yellow, 40 for the blue. Those are the magnifications. Now after that, I also see it's 4/10.10, 10/0.25, right? 40/0.65. They've already figured out the numerical aperture, so that was nice. But I wanted us to understand how we got there. So those numbers are usually written on the objectives too. And then there's oftentimes some other objective-specific information, like maybe the tube length of the objective and slash the thickness of the cover slip that should be used. So that's shown here. So there can be other information put on the objectives too. Now that brings us all back to the resolution of light microscopy. Why is it that we are limited in what we can resolve? So the resolution of light microscopy is D in this equation, right? So it's equal to a constant, 0.61, times the wavelength of light, right, divided by the NA. And we've gone through kind of understanding the NA, and that's specific for certain objectives. So the shortest wavelength of light that we can see, right, violet, is about 400 nanometers. Okay, that's fine. So now let's think about a standard 63 times lens. And it's written right on there that it has an NA of 1.4. So we're kind of given that. We could just throw that values into the resolution equation, right? So D would be 0.61, that constant, times 400 nanometers divided by 1.4, the NA. And that gives us 174 nanometers. Or, right, the minimal distance that you would need to see before, in between two spots to be able to resolve them. Or to put it another way, using this setup, if there's two spots in space that are closer than 174 nanometers, to the eye, it's going to look like one spot, right? They can't be resolved. So that's a good number that I just have in my mind. It's one of the numbers that I think is important enough that I'm sure that I remember it, right? And it's 200 nanometers. And that's about the typical limit of light resolution, of light microscopy and what it can resolve. Right, so there's two key equations here. We talked about numerical aperture, and that's dependent on the objective, because the objective strength, right, is going to be related to the mu, right, or how close to the sample it needs to be for magnification. Right? And don't forget N, though, because we can also think about air versus oil, for instance, with refractive indices in between the objective and our sample. And then the other one is the one we just looked at, right, the limits of resolution, and throwing in some pretty standard examples of a type of microscope we might be using. That gets us to about 200 nanometers, or about the limit of resolution. And to look in the spectrum of life, right, so this illustrator apparently has thrown in a guitar as something that's a meter compared to a carbon atom. We can see a variety of different biological structures here, right? A cell, and when I think cell, I'm usually thinking like about 50 micrometers, a standard eukaryotic cell, down to an E. coli bacterium cell, right? So this is prokaryotic. Normally when I'm thinking prokaryotes, I'm thinking about a micrometer. I mean, of course, there's going to be some variation, but these are kind of what I have in my own mind as go-to numbers if I'm thinking about context. And then below that, we have organelles like ribosomes. And so now the illustrators put on our light microscopy as being, useful. About a millimeter is about right. So that's the biggest thing I would use for transmission electron microscopy, using a scanning electron or a scanning objective would be about a millimeter. You may have encountered classroom lessons where the students are asked to first start working with a microscope and they put a ruler under it, right? A millimeter is a good kind of starting point on getting them comfortable with what a scanning objective is useful for. But then theoretically, right, it can go down to 0.2 micrometers or 200 nanometers. I don't know why the illustrator here has gone as far as 0.1, because that's about half the distance as I think is usually accepted. But nevertheless, that's about the spectrum of what we can do with light microscopy. Now, electron microscopes we'll look at coming up. They for sure can beat light microscopes in terms of powers of magnification and resolution, for sure. They can see much smaller stuff. But that doesn't mean they're only good for that. So there's overlap there because they're also useful for seeing different types of structures that you can see with light microscopy too, just different types of information. So I talked about Namarsky DIC as a variation on light microscopes. I just want to point out that these are three different structures that are using Namarsky differential interference contrast. So the light is actually split by a crystal, goes through a sample, and then is recombined by a crystal or a prism. And when that light is split, it experiences different parts of the sample in different densities, so it gets knocked out of phase. And when it's recombined, that influences whether it gets like brighter or darker. So it's a way that we can manipulate light that provides information on the different material properties of the samples that it goes through. And what you'll see is it looks a little like, I think about it like a moonscape, right? It looks almost like the surface of the moon with grayscale darks to lights, and it looks very three-dimensional, like it's popping out. This is Namarsky DIC, so it gives you that additional kind of contrast. And phase contrast, looking at the same samples, is actually more standard. A lot of teaching microscopes have this ability, but it also influences a path of light to bring out and introduce contrast where it's not normally present. Comparing bright field and fluorescence microscopy, so to the left is bright field. You'll notice that there's a light source on the bottom. It goes through your very thin sample and then up towards the eyepiece, right? And we talked about how different objectives can be used to get increasing magnification and resolution. A fluorescence microscope is shown on the right, and it behaves very, very similarly. The only difference is that if you remember, something that's fluorescent needs to get excited by a certain wavelength, and then it emits light in a certain wavelength. And those wavelengths are different because energy is lost in that transfer. So in this case here, right, the sample needs to be bathed in blue light. So there's some sort of a light source, right? It could be a lamp, or shown here as a laser. And it comes in and it excites with a specific wavelength. And you can use filters to kind of dial in the light that's going into your sample to make sure you're just exciting what you want to excite with the appropriate wavelength. And then there's light emitted from your sample, right? So in this case here, the fluorophores were excited by blue light, and now they emit green light. So now the green is leaving your sample, and it follows the typical path through the optics up to the eyepiece. But you can, again, throw in some filters and mirrors to dial in and kind of polish the light going in and the light coming out, so that you don't get like this noise outside of the sample that you're interested in. Because if you remember, some things are kind of autofluorescent and some things get excited and emit light unexpectedly. So you can really kind of fine tune it just for your sample. So looking at some different comparisons, right, here's bright field. So this is just standard light through a sample. The sample here is algae. So it doesn't need to be processed or sectioned like a very fine deli slicer because it's already so small, it's transparent. And it has some just natural color, right? So it has chlorophyll. We can see that it has green. But in that second panel with the fluorescence, the investigators have added something that looks like it stains the cell wall. The green, a little tough to tell without knowing more information. Tough to tell. I don't know what it's staining, but some sort of subcellular structure that they want to specifically identify, right? So we looked at the advantages of fluorescence microscopy, that you can come in and highlight a specific protein or a specific structure that you can't see on the left here, right? You can see beautiful cellular structure, but you can't tease out like individual players and biochemical cascades and things like that. But with fluorescence microscopy, you can use those stains, and you can do that. Now with electron microscopy, which we'll look at coming up, again, there's some electron microscopy that behaves like bright field where light passes through it, and those sections need to be thin. And these are all on the same type of single cellular algae. And this also highlights that it's not necessarily always about magnification, although electron microscopy for sure can magnify more. That's not always the point. Because you'll see on the left, the transmission electron microscopy has some sort of stains that were applied so that usually heavy metals, right, are sticking to some structures. So it looks like they're actually sticking to some of the lamellar membranous structures here. So maybe it's some sort of a lipophilic or a lipid stain. But it's identifying and highlighting some organelles and things that you couldn't see with a light microscope. Now to the right, notice that this looks more like where the electrons are coming down from on top. You're not looking at electrons coming through, but you're looking at topological kind of structures. This is like the comparison between transmission and scanning in stereo microscopy, right? Where you need light going through or light coming from above. Same thing with the TEM is transmission electron microscopy, electrons coming through. And scanning electron microscopy is like is the electrons being cast down from above, and what you're detecting or looking at is the way that they scatter off of your material so that you have kind of three-dimensional physical shape information. Right, so here on the left is where you have a source of electrons. So it's usually a coil that has kind of power run through it, and the coil emits electrons. And they go through your sample. So this is the transmission electron microscopy. It usually requires a lot more work than light microscopy, where you have to fix the samples and you are cutting them super thin. So usually cutting them with like diamond knives or specially kind of created glass knives, or very fine knives to take unimaginably thin slices of a material, and that ends up in your sample. And again, you also have to stain it usually with some electron dense stain that sticks to materials that you're interested in, because as that electron beam hits the sample, right, some electrons will go through and give a lot of signal, but some areas of your sample will be made to be electron dense and block the electrons. So there'll be less of them to go through to the detector, and that's how you see your image. And when I first was doing this microscopy back in the early 2000s at UMass, there were these electron microscopes in the basement of the science building there. There are these old workhorses from like the 1960s and 70s. These microscopes were very, very mechanical, so there wasn't a lot in the way of electronics or computers. So they're all like, there's almost like a 1950s Ford or something like that. All the pieces were very apparent and clear. And the electrons would actually go through, and the detector was photographic film that I would put under there. I'd put a sheet of photographic film and just count 1, 2, 3, 4, 5, and then bring the film to the darkroom. If it was too bright, then I'd have to adjust, or too dark, I'd have to adjust how long I exposed for. But it was all very, very direct and analog. Now the detectors these days are, computerized, so everything's being detected by detectors that are actually receiving the photons and translating that to a digital image. Now to the right is I think more fun electron microscopy, because you're not limited by these long sample preps and making things very thin. But you can put kind of whatever you want on that stage, like a whole fruit fly, within reason. Sometimes those have to be treated. Like if you were to take a fruit fly, it might work if you just threw it under there, but you're usually better off dehydrating it, because it ends up having to all of this has to happen in a vacuum, because everything else would introduce noise. So dehydrating it makes it resistant to the vacuum. And then you come in with something called a sputter coater, which basically coats your sample in a thin layer of gold, and that's electron dense, right? So then electrons are really going to bounce off your specimen because it's been coated in some sort of a metal. But that's what you would put on the sample, right? Your electron beams are cast down from above, and then they scatter based on the shape of the material, right? The detector can then interpret that, right? So you might think about like if you were to cast down signal upon a street with drains and like a manhole, and a cone was on the street, right? Electrons that went down into like the open manhole would disappear. So those would be kind of lost and gone from a detector and would show up like a dark hole. Whereas electrons that struck a side of the cone and ricocheted off directly into a detector would be super bright, and that would be reflected with your detector as an intense area. Whereas ones off to the side might go towards a detector but might not hit it head on, would give kind of grayscale or intermediate signal. So it's a way that those electrons are scattering and being detected that gives information on the shape of something. All right, so here's the TEM, transmission electron microscopy, very powerful, 70,000 times, and an awful lot of information there. And then these are samples of scanning electron microscopy images. So there's some HeLa cells, if you're familiar with the Henrietta Lacks story of the cancer cells. Those are examples there on the left. Powdery mildew, and then Arabidopsis is very common. Our fluorescence talk from last week was relative to the tobacco plant. I was thinking maybe it was Arabidopsis because it's such a common plant science model system, but that's shown there in C. And to summarize and just look at it all together, right, these are the different types of microscopy. So we looked at how you can bring to light things that are not normally resolvable with the unaided eye. Right, so with light microscopy and electron microscopy, both of those approaches that we see there on top are bringing down some sort of particles, right, photons or electrons from the top, and then visualizing the way they bounce off of or interact with your sample. Whereas on the bottom two panels, right, it's either light or electrons passing through your sample. We looked at some of the limits to light microscopy and how that's dependent on objectives, and how more important than magnification really is resolution. Magnification, like, kind of who cares? What you need is for data, for information. What you need is resolution. And then on the lower right, those are electrons instead passing through a sample. For sure, the investigators here have thrown on some osmium tetroxide or some sort of toxic, nasty heavy metals that are sticking to tissue structures that are more electron dense or more opaque, and that comes out as lack of signal. Okay, so we will catch up next week, where we'll move on to the next kind of content area, and this is all building together towards an integrated STEM project that we'll kind of go through. And then you'll be off to think about some integrated STEM projects and how you might like those to take shape. Thanks.